great that people are getting lunch, and I think we should just see this kind of like as a evening at the cinema. You know, you're getting your popcorn, you're going to enjoy a great session here. I'm going to go ahead and get started because I would really like to preserve some time for some back and forth conversation with Desiree. So first, good afternoon. We're now 5 minutes into the afternoon. And thanks for joining here at the Global Health Research Spotlight. This is hosted by the Center for Innovation in Global Health. This series showcases outstanding global health research being led by Stanford researchers. My name is Steve Luby. I'm the Director of Research for the Center for Innovation and Global Health. And before we get started, I want to tell you about two upcoming opportunities. Ola in the back will reinforce this if you want to hear anything more about it. But we're going to have a Global Health Research Methods Workshop on December 8 and December 10. And this is particularly focused on fellows, residents, and even faculty in the School of Medicine. So apply by October 26th. And in January 28th, we're going to have the first ever combined Center for Innovation in Global Health, Center for Human and Planetary Health research convening. So we are looking forward to that, and we are still seeking abstract. The abstract deadline is October 23rd, and I realize that most of you plan your abstracts weeks and months ahead, but for those of you who tend to get in at the last minute is approaching. I want to introduce our speaker for today, who's my friend and my colleague, Desiree LaBeaud. Desiree is a physician scientist. She's a professor in pediatric infectious disease, and she's the associate dean of global health research here at Stanford in the Center for Innovation and Global Health. She's also a senior fellow at the Woods Institute for the Environment, and you'll see this work between her physician's perspective and the environment perspective in her work. Much of her work historically has focused on the epidemiology and ecology of mosquito-transmitted viruses, but more recently she's been working more on the global plastic pollution crisis. Desiree is passionate. She's passionate about many things, but definitely passionate about global health and global justice, and her work is really community engaged, focused on long-term partnerships. She works with in-country collaborators in Kenya, Grenada, Pakistan, and Brazil. Not only did she run a panel yesterday on community engaged partnership, she co-edited a book on community engaged partnerships. She received her MD from the Medical College of Wisconsin and earned her master's degree in clinical research and epidemiology at Case Western. Today she's going to talk about her community engaged arboviral research and how this has sparked her new efforts to foster environmental justice. So Desiree, over to you.
Thank you. And imagine everybody can hear me. Can you hear me? Okay, that's good. Okay, great. Thanks so much for coming. And thanks for the introduction, Steve. I'm going to run you through our work, really our recent work. I tried to keep it to really like the last 5 years or so. And it'll be a lot of things you haven't seen, I think. So stay tuned. And I'm hoping that you kind of see the arc of our work. I called it From Disease to Justice. This is what we're pressing for in our work. So I thought I'd start at the beginning. So when I was a pediatric resident, I went to Rainbow Babies and Children's Hospital because at the time it was one of 2 programs in the country that had an international health track. Now they all do. And during my rotation, my international health track, I spent time working in Laos. And I happened to go to Vientiane during monsoon season, and I saw a lot of children die of dengue. And it really impacted me, and I came face to face with the fact that mosquitoes are the most deadly animals in the world. And I went back to Case and thought, well, maybe if I get some infectious disease training, I might be like slightly useful in the global setting. So I went back, I did my infectious disease fellowship, and then got my master's in epi, and then came to California in 2009, and came to Stanford about 11 years ago. And all this time, I was working in Kenya. So like, I love Kenya. I know Kenya's my heart. And when I started working in Kenya, I was working in really rural areas. I was working in what was called at the time Northeast Province on the Somali border. And I was working with a wonderful mentor, Charlie King, who continues to be my mentor today. And Charlie was actually a schistosomiasis expert. He is world renowned for schisto and schisto work. But he had this NIH biodefense supplement on something called Rift Valley fever, which he didn't know anything about, and neither did I. And so we both sort of jumped in together, and I fell in love with what remains my most favorite arbovirus, which is Rift Valley fever. So for those of you who don't know, Rift is like the quintessential One Health pathogen because it impacts animal species and human species. In humans, it's a significant threat to human health because it can cause hemorrhagic fever and encephalitis and blindness and really a lot of long-standing health complications. It was initially discovered in the early 1900s in the beautiful Great Rift Valley of Kenya during a large sheep epizootic. And it has a really interesting life cycle that is super climate connected because outbreaks of Rift usually begin after heavy seasonal or upland rains or ENSO events where shallow depressions in the landscape called dambos fill with water. And in these dambos are these Aedes resistant, or these desiccation resistant bloodwater Aedes eggs. And so they're already infected with virus. So when the rains come, you get 1 million mosquitoes all at once, and they all have virus. And then the ruminants, the livestock, come to the dambos to drink water, and these floodwater Aedes prefer to breed and bite furry beasts. And so they bite these ruminants. The ruminants get really, really high virus. They're amplifying hosts for the virus, tons of virus. And then secondary vectors like Culex mosquitoes that are more promiscuous feeders then bite them and then can bite us and spread infection. Or humans can be infected with direct interaction with infected livestock and through animal husbandry activities. And so, during those years, I came face to face with environmental change and these diseases and truly in poverty, right? And found that extreme weather events are really linked to disease. This is just one homestead we were working with, and this is it during the rainy season. You can see how it greens up and how things change. And so Rift really led me to the interconnected web of arbovirology that I hold so dear. I think arbovirology is the greatest system to study for anybody looking for a system because it's so interesting. Variety is the spice of life, and look at all you get to do. You can do virology, you can do vector biology, you can do human and animal medicine, and then there's all the intersections, right? Ecology, epidemiology, human and animal behavior, immunology, genetics, all in the background of environment, right? And so it really brings everything together, and you get a very holistic view of this disease system, and that's what I'm hoping to show you today when I talk about what we've been doing in Kenya. Now, I've been working in Kenya for 2 decades, but like I said, I'm going to try and focus on just the last few years here. And I am a spokesperson today talking about this work, but I am in no way the person behind all of this work. I was, a collaborator, a mentor, a cheerleader, maybe a thought partner in a lot of the work. But it's taken so many amazing people, and I've had the good fortune and honor to work with so many just brilliant scientists, field workers, community members, policymakers. It's just been such a fun journey. And I have an incredible team here at Stanford as well, which you'll see on my joy slide later. And then I just want to thank our funders, some of which are in the room, Human Planetary Health and the Fogarty GHES. And then I couldn't go on without calling out my 2 long-standing scientific partners in Kenya, Bryson Ndenga from the Kenya Medical Research Institute and Francis Matuku from Technical University of Mombasa. All right, so just some recent things that we've done with Rift. So I studied Rift for many, many, many years, and I had the good fortune of doing this very large systematic lit review funded by CEPI to look at really the remaining gaps in Rift epidemiology, and there still are many, despite people working on this. And the fun thing about this was that it was 3 generations of people who love Rift. So there was me, there was my mentor, Charlie, and then there was my mentee, Kelly Gherkin. And Kelly is a veterinarian who was with me as a GHES postdoc for a few years. She's now at Liverpool getting her PhD in Rift, which is amazing. But Kelly had interest in urban Rift Valley fever. You know, most of what we know about Rift Valley fever is through our understanding of sort of rural disease risk and all of those animal husbandry activities. But what about urban settings where most of the people are? Is this a big problem where Rift is endemic in Kenya? And so Kelly worked a lot on urban Rift and realized, you know, the risk is low, but it's actually independent of livestock ownership. It's not the disease that we think of in the rural settings. And that people know about Rift, but the high-risk groups really recognize their vulnerability because of the urban livestock systems. And this is both movement of livestock and movement of livestock products like milk, which we have really weak safeguards for. Kelly did a lot of work in abattoirs and with the milk and meat trade to discover all of this. And so for a long time, I had been interested in milk as a pediatrician. And we had a lot of epidemiologic papers to kind of associate raw milk exposure and Rift Valley fever. But we also knew through Kelly's work that milk handling consumption was a significant risk factor. And recent studies in the last couple of years have for the first time really identified the virus during outbreaks in cow's milk. And so the pending and looming question is, well, there's this association, but you know, is it even possible for milk to really remain infectious in milk? And so Brian, who's sitting right over there, who came to our lab as a resident and then stayed on as an ID fellow and is currently an ID fellow in adult ID, has a PhD in virology and has worked with high containment viruses in the past and did some amazing work looking at Rift Valley fever stability in milk. And what Brian did was he mirrored the conditions that Kelly had figured out in the milk trade in Kenya with those big silver containers on the backs of the bodas, right, measuring the temperature. And so he did work with Rift at refrigerated temperatures and then field temperatures of 25 and even the hot field temperatures of 34 degrees to see if Rift was stable. And it turns out it was. It remained stable and infectious for 4 days when it was cold, 2 days when it was hot. But luckily, pasteurization works, flash and regular pasteurization. So thank God for this, actually. But the issue is in our setting, a lot of people aren't boiling their milk. They're expecting the other person to have boiled it. And so it doesn't happen. And so people are still at risk. And Brian showed that, you know, you can PCR the milk even after it's been pasteurized and you still detect RVF RNA. And so together, these findings really suggest that milk might be this really important potential public health hazard. Boiling works, but people need to do it, and PCR screening might be great for surveillance. And so USDA is very interested in this and continues to fund this work, and we'll keep going with it. So now I'm going to move on to some other viruses. So along the way, I've integrated a lot of other mosquito-borne viruses into our work, and we worked for about 10 years in 4 locations in Kenya. 2 on the west side, on the beautiful shores of Lake Victoria, Kisumu being Kenya's 3rd largest city, in Chilambo, a rural area, and then on the beautiful Indian Ocean coast with Ukunda as an urban area, and Msambweni as a rural area. And for a little over 10 years until this current presidency, we had a large NIH-funded project to really understand and confirm the burden of these infections, determine the proportion of febrile illness due to them, understand disease complications, document all of the vector ecology work, and infection work, abundance work, and then link all of that to climate. And so, you know, I'm there in Kenya, you know, suffering mosquito bites like everyone else. And still, during this time, over the years, when you get bitten by mosquitoes and you get a fever, it still is assumed most often that what you have happening to you is malaria. And this runs deep. And but the point is like, what if that fever isn't malaria? And you know, what if you're actually getting bitten by a day-biting mosquito, which you're not protected from when you're underneath that net? You know, and I can't believe this, but I always have to keep this picture on my slide. These are my 3 sons 10 years ago. Ah! Okay, so anyway, they can't fit under the same net anymore. So, like, what are these other vector-borne diseases other than malaria actually common and important, right? And so we set off on trying to understand this. And so there are folks in my lab, like Ezra, who runs our lab, and Bethel, who was a Stanford undergrad and then went on to be a lab tech and now is a UCLA med student, have looked at, what's happening with our populations, how often are they getting exposed to dengue and chikungunya. You can see these are just survival curves, so as the line goes down, that means more and more people are getting infected. And you can see it's just kind of a steady trickle. What we're looking at here is, again, Akunda on the coast and Kisumu on the west side. And when you look overall, it seems that dengue is a little bit more common on the coast, with about half of people being exposed, and chikungunya is about a quarter of people exposed in both places. And if you look over the age ranges, as you'd expect, looking at seroprevalence, the older a person is, the more likely are to become face-to-face with an infected vector, right? But there's still a lot of kids being exposed, 10 to 20% of kids, so that's a lot of kids. We've looked at risk factors, and what we find time and time again is that whether or not you're looking at just exposure or recent infection, which is the bottom chunk, wealth and, of course, the built environment really matter, right? The poorer you are, the more likely you are to get these infections. And if you have, water that you're holding because you don't have safe, reliable access to clean water, like a quarter of the world doesn't, you're going to be more likely to be exposed. So that's fine. Lots of exposure, fine. But what about disease, right? We know that arboviruses in particular, a lot of them cause a lot of asymptomatic infections. So that means you get the virus, but you don't show any disease from it. Are kids actually getting sick? Well, Melissa, who actually just visited our lab yesterday, she's now at CDC, but she was an ID fellow in our lab. She looked at some of our samples. So we had 1,000 kids or so coming in with fever. And she PCR'd all of them to see, well, how many of them have dengue in their blood. And we were shocked to find in this subset of samples that 40% of fever, 40% of kids with fever had dengue. This is like astonishing. For an infection that no one knows about, no one's testing for in the health center, there's no vector control for it. We found all 4 serotypes, so hyperendemic dengue in Kenya. And the most probably important finding was that half of these kids were actually smear positive for malaria, right? So they would have all been called malaria. And so we've gone on to sequence this. I had a PhD student for 5 years in my lab, Cindiso, an epi student who looked at the molecular epidemiology of these viruses and asked, where are these outbreaks coming from? And so we did a lot of sequencing and phylogenetic analysis and found that there are a lot of distinct lineages and that these lineages are consistently and repeatedly being introduced from East and Southeast Asia over the last 10 to 20 years. There's a huge connection between Asia and Africa. And clearly, these infections are not in isolation, right? There's a lot of arbovirus and malaria co-circulation and co-infection. And what we're just showing here, you don't really need to look at all the numbers, but just trust me that it happens that you get acute dengue plus chick, or chick plus malaria, or malaria plus dengue, or all 3. It happens, right? And, it makes sense because these infections are actually overlapping in space and time in our study sites, as you can see in these maps that Aslam, who is another PhD ID fellow in my lab, completed with Donald, who's a spatial epidemiologist we like to work with. And again, poverty is important, and we started to see more and more in our work that the presence of litter around a homestead really was an important risk factor for being exposed to these infections. So what do the infections look like clinically, right? It turns out that they all look the same, right? In these graphs, what you're looking at here are, in blue is dengue only, in green is dengue plus malaria, in orange is malaria only. And you can see that the symptoms are completely overlapping. And David is really interested in co-infection. He's been with me actually before my Stanford days. He came with me to Stanford many years back. But nor man nor machine can actually figure this out. He did machine learning models to see if they could outcompete, clinicians in diagnosing and distinguishing these infections. And it's impossible to do so, right? Because again, in the clinics where we're working in Kenya, the one test that we really have to run is a malaria smear or malaria RDT. And dengue in most of the world, it's actually clinical diagnosis. And the way people diagnose dengue is not necessarily through blood tests, although that's confirmatory, but it's through clinical diagnosis with a cluster of symptoms, right? The WHO actually puts out what these symptoms are. And so Aslam, again, in my lab, he wanted to see, well, do the infections in Kenya actually, can they be confirmed and clinically diagnosed using those WHO criteria? And so he looked at our study set, and it turns out that if you use WHO criteria in Kenya to try and diagnose dengue, which is all you've got because there's no testing, you miss 62% of dengue cases. This is terrible news for us, right? It turns out that although children with dengue in Kenya are less severe than other parts of the world, they can still have a lot of severe symptoms, but they're being missed by the current guidelines. And so what does that lead to? It leads to a lot of malaria overdiagnosis and a lot of antibiotic overuse. I know there's a lot of SASH scholars in the room. There have been a lot of SASH scholars that are really thinking about antibiotics. But Annika, who is a different peds resident in my lab, looked at malaria diagnosis and just antibiotic overuse, and there's just a lot. And Melanie, more recently, looked at more recent studies or more recent cohort studies and found that antibiotics were still prescribed in about 73% of encounters, and actually being a child puts you more at risk for getting antibiotics. And if you're malaria negative, you're 4 times more likely to get antibiotics, right? And all of this makes sense when you're limited in what you can rule in your infections. And the infections and the consequences don't just stop with kids or antibiotics, because we've also looked at impacts in pregnancy. So we know from systematic reviews that severe dengue, hemorrhagic dengue, can be very severe in pregnancy. It can lead to abruption, stillbirth, low birth weight, and so forth, prematurity. But we wanted to look in our milder dengue infections in Kenya, is this impacting pregnant women at all? And so we had a prospective cohort study where we measured women when they came in for their first antenatal visit and then at delivery, and looked for those who seroconverted to dengue, meaning that they had been exposed during their pregnancy. And we were shocked. We expected about a 4% incidence rate, because that's what we had seen in our other studies. But these pregnant women, about 12% of them actually seroconverted for dengue, which was a lot more than we were expecting. So maybe there's something particular about that movement or where those women were that put them at risk. In this study, the way the study was designed, we could not keep very premature babies or, of course, stillbirths and things in our studies, so there is a little bit of a potential type B error. But we did find, even with mild dengue, an association with potential low birth weight. So interesting to think about. And interesting to continue to think about what the consequences of health are in Kenya for these infections. And then, just one more stunner before I move on to kind of a different thing. So I've showed you a lot of seroprevalence, right? I've showed you, wow, so many people exposed, 50%, 20% of kids, it's a lot, right? Well, our recent study also shows that even what we're doing with that is a huge underestimation, because there's actually a lack of seroconversion in our viremic patients. And so, David, we have in this study, say 8,000 people coming with febrile illness. About half of them had dengue PCR results, and about 1/10 of those were positive. Okay, 10% dengue positive. A lot of disease, again, a lot of incident disease for nothing that's being monitored. Okay, so of those 475, 428, this was their very first dengue infection. They were IgG negative at the time of their PCR diagnosis, right? So what you would expect is at the 4 to 6 week follow-up convalescent visit that all of those people, recognizing that we found dengue in their blood, would seroconvert, right? But what we found instead was that 290 failed to seroconvert. And so this is a lot. That is a huge percentage. That sort of doesn't go along with our paradigm of how we think human immunity works. And we continue to think about this, ponder it, and work on it, and we will publish it. And we can have a conversation about this at the end. Okay, so this is a complex system, right? And there are a lot of challenges where I'm working in Kenya. We have a lot of sensitive and vulnerable populations, a lot of widespread competent mosquito vectors, limited physician knowledge and clinical suspicion, poor diagnostics, no accurate, rapid local testing, not any available treatments, vaccines, no current vector control for these infections, and a shifting climate. And so I just want to briefly touch on the shifting climate, because we've done a lot of work recently on this. What are the effects of climate change on these systems? Well, we know that each vector has a temperature optimum by which its biology works best, right? So the malaria vector functions best around 25 degrees, and the dengue vector likes it hotter around 29 degrees. And so Melissa said, okay, that's fine. We have these ecological models, but does this really matter for the clinics? What's happening in the clinics? And she measured the effect of 30-day mean temperature on smear positivity at our 4 clinical sites, the ones that I showed you previously. And controlling for rainfall, bed net use, sex, age, and socioeconomic status, what she found was that malaria positivity in the clinics actually peaked at 25 degrees Celsius, completely mirroring the ecological models, which was very interesting. So when you overlap that, and you look at malaria and non-malarial illness, you'll see there is a lot of overlap. But as the world warms, there's more and more non-malarial illness. And when you take a step back and you think about that in Africa as a whole, malaria burden is going to shift as things warm across the continent, but dengue burden is just going to grow and grow and grow, and we are unprepared, right? Like I said, with those challenges, there's a lot of work to do to protect people from any kind of consequences from these infections. So that's temperature. We've also looked at other extreme climate impacts. Cameron was an undergraduate in the lab who looked at satellite-derived climate data and our ground truth data, looking at floods and floods and heat waves and cold waves, and looked at vector abundance in cases of dengue infection following these climate extremes. And what he found was that floods were significantly associated with vector abundance and higher risk of dengue. And this, of course, makes sense because you need water for the life cycle of a mosquito, right? But we've gone on to really think deeply about this and have modeled climate and its impacts on vector-borne diseases in multiple equatorial countries, and seen that it's really, there's this non-linear model and relationship between rainfall and vector abundance, where when you actually have drought, you're also likely to have these vectors, not because things are dried up, but because people don't have safe, reliable access to clean water, and they have these containers that then become mosquito breeding factories, right? And then when it rains, out in the community, you have a lot of waste or trash that then fills up and becomes breeding sites, which I will talk about in a little bit. And so again, the underlying inequities, poverty, and inability to have reliable access to safe, clean water come up as huge risks for these diseases. So what are we going to do about this bad girl, right? Now that I've depressed you, and she's wreaking a lot of havoc on our populations, right? We need to control her. And the only way we can really prevent human infection is to prevent the bite of her. So how do we do that? Well, a lot of the ways we do it, and some of the studies that I'll show you, we intervene at the household level because that's what people have control over, right? But we have done some work more recently looking at the non-household level because she is a day biter, and you're not always at home during the day, right? So Victor in my lab, who's a modeler and a postdoc, figured out that more than half of the infections happening in our environments are actually happening in non-household environments, right? So it's not just about the household. We have to include non-household environments when we think about control. And I'll just point you to this. What you want to see is darker colors, because darker colors means more control. It's really important that we do non-household control, both before and as a response to epidemics. Okay, so time to intervene. We initially, I've been was NIH funded for like 20 years, and it's really difficult to do clinical trials with NIH funding. It's like a whole problem. And so when I moved to Stanford, I was like, woo, we can do intervention studies with Stanford funded dollars. And so that's what we did. So I got here, we started doing that. And I started working with Jenna Forsyth, who Steve knows very well, who is an EIPER PhD student. And we spent a whole year in Kenya understanding what people know about these vector-borne diseases. And as you would expect, everybody knows about malaria. They know about the night-biting mosquito. They know they need to sleep under the net. But less is known about these other mosquitoes, day-biting mosquitoes, and the fact that you can clean up, the environment around you and really protect against familial risk. And then, the two Kenyans that I said that were so important to me, Francis and Bryson, they're both medical entomologists. So we have a lot of medical entomology in our work. And it turned out that water purpose really mattered. We found a lot of these mosquitoes breeding in so many different containers. You can see them there. And when you work with a lot of entomologists, a lot of the lore is that you talk about the specific container, like tires are very bad, or, these jerry cans are very bad, right? But actually, we found in our work that it wasn't about the container. It was about the purpose of the water in the container. And that makes perfect sense because it takes about a week for these mosquitoes to develop. So even if you have a completely unsafe container sitting around breeding mosquitoes, if you're using it a lot because you're washing with it or it's for sanitation or whatever, it goes, right? But it's that non-purposed water. It's the purpose that's the water that's just sitting around in containers that have no purpose that end up breeding the biggest majority, 81% of our larvae. So Jen and I got together and we were like, let's design an intervention study to deal with this problem. So we wanted to understand the barriers and facilitators to household source reduction, and we developed this school and community-based intervention. We trialed it out in the settings. We improved it, reiterated the design to promote more facilitators of household source reduction. And there were really 3 parts to our intervention. We went home to home and worked with community members. We went into schools and taught kids all about mosquitoes and disease and prevention. And then we ended up having a huge community tree planting and cleanup. And I can tell you there's many papers about like the knowledge and attitudes and practices and how it was sustainable over time and all of that. But what was most interesting about this study is that, and I should just say, but it was good work and it was done by Pratik, who was a Rice student, who's now gone on to be a med student at Baylor. But we really dug in and wanted the kids to feel like they were agents of change. So we made them these mosquito scouts. We gave them badges. We taught them games, this mosquito tag game all about the mosquito life cycle. And we gave them a lot of homework. And the first day, we showed them what immature mosquitoes look like, these little wormy things in the water, and told them to go out and find that breeding find that in their neighborhoods. And they all come back saying, yeah, we found it breeding in all the plastic trash behind our homes. And so that's when we decided to do a big community cleanup. And these 250 school kids over 1 month picked up 17,000 plastic containers, more than a ton of plastic waste. And we tried to repurpose as much of that waste as we possibly could. We planted 4,000 native tree seedlings in the schoolyards and the homesteads of the kids. So we have these nice little forests to go back to now when we visit Kenya. But we had a ton of plastic waste to deal with, and we could either burn it or bury it. So we buried it. And that brings me to the bit that Steve highlighted that I would be talking about, which is plastic pollution and vector-borne diseases. So everyone in the room knows that the plastic crisis is an exponential one that is going, big oil is making money off of this, and so it's going to keep going up exponentially unless we do something about it. There's more than 9 billion tons of plastic waste already on our planet. And again, 40% of it is single-use plastic. And our work, mainly done by this amazing individual, Njenga Ngugi, who was our PhD student at Chuka University, we really figured out over time that plastic trash and plastic waste was what was breeding most of our mosquitoes. He captured over 2 years more than 22,000 samples and found them breeding in these huge piles of trash in our neighborhoods, which were really close to human dwellings, and leading to 50% of our communities being exposed to these viruses. So I'm an optimist. So there's an opportunity here, right? If we have all this trash laying around, breeding mosquitoes and making people sick, if we did something about it, maybe we could prevent disease. And it brings me to the circular economies of waste, right? Not only could we prevent disease, but perhaps we could also improve the 40% underemployment in our area while we do it at the same time. And so that's what we did. We started a business incubation program among these yard shop operators, which were these folks who were sort of in the informal plastic recycling trade. And we improved their business skills and strengthened their relationships with key stakeholders like the government and each other. And then also provided training on health and safety and waste sorting practices, because they're just trying to make a living, but how they do it can either make or break the health of their community. And we wanted them to feel like there was something else other than just economy, but actually health and sustainability that was important. And if they were able to sort and protect their waste, they were actually heroes for their community because they were getting it off the streets. And so over time, this led to an increase in the amount of plastic waste being traded, so more money for them, less fluctuations in prices, a really strong peer network, and significant reductions in the breeding of the mosquito, which was all good news. But during this work for the 5 years, we realized there was a lot more to do. We needed to create more community awareness. We needed to improve waste infrastructure, create a market for second-use products, work with our policymakers to really change policy and enforce policies, and then cultivate a recycling culture in the community. And so to do all that, Francis, Bryson, and I launched a nonprofit 4 years ago called the Health and Environmental Research Institute. And we call it HARI for short, which means blessings in Swahili. And what we do at HARI is really try to inspire collective action among scientists and community members and policymakers to improve the trajectory of health for the planet and its people. So we have an educational hub, we're changing social norms, we're destigmatizing trash collection, we're doing a lot of community relationship strengthening, creating recycling programs and jobs, and then decreasing these mosquito and trash-borne health risks. These were the original 25 founding members. There's Francis in the center, looking very proud. And so what we've been doing lately, in my last couple of minutes here, I'll just say to you, you know, plastic is a big thing, and we're always working on plastic, but actually green waste, food waste, compostable waste also is a problem because it breeds a lot of rodents, leptospirosis, bacteria, also creates habitat for mosquitoes. And what we found when we did that study, the Trash to Treasure study, where we worked with yard shop operators, is that there were no women. There were just very few women in waste. And so again, we had an opportunity to create a circular economy. So this year in January, we launched a black soldier fly farm, which is a nature-based solution to deal with waste. And they are amazing. I have very cute pictures of these things if you want to come see. They're amazing little beasts. So you rear the adults, but the amazing creatures are the babies, right? The larvae, which just eat, it's like the hungry caterpillar. They just eat compostable waste. And when they do that, they produce two products. They poop, which is frass, which is an incredible fertilizer. And then there's their bodies, which is the expensive protein component of chicken and livestock feed. And so you take something like waste, which is breeding rodents and methane and mosquitoes, and then you turn it into food and fertilizer and jobs for women. And so that's what we've done. Meet Carol, our black soldier fly farm lead. A lot of this work is also done with Amelia in my lab. And we're making stuff. I mean, these are our love cages. Look how happy they are. Those adult flies are making babies. We call them the love cages. We play them music. And then we're making fertilizer. We're making food. We are measuring greenhouse gas emissions, because again, methane, like waste just sitting around creates a lot of methane. So we're doing all of that. We're doing LCA analysis and showing that this is a very sustainable way to process waste. And so, women-run black soldier fly farming reduces disease and environmental impact, provides jobs for women, creates economically valuable products more sustainably, and improves food security and nutrition. And I don't have the time to show you all this work we've done in Kenya, but I've been working here a long time, also on child nutrition, and about 25% of the kids where I work are undernourished, about 12% are stunted. So it's really important to get food. My dream that I can tell all of you is that when we really hit sustainability or making like a ton of chicken and eggs, I want to donate all the eggs to the school food program because I think it's like an egg is just a perfect source of nutrition, right? Yeah. The other thing we're doing, so the other thing is, again, building awareness, right? So I've gotten the opportunity to work with an incredible Brooklyn-based artist named Jean Shin, and we got a grant to work with HARI and a lot of the local universities in Kenya on the South Coast, a ton of students, to build this gigantic sculpture, which sat for a year from Earth Day 2023 to 2024 in the center of town. Just, again, building awareness and starting dialogue, kind of like we did yesterday about partnership, right? About waste and plastic and its impacts on the ocean. Because again, remember, this is just about half a mile from the beautiful Indian Ocean coast. And the underlying problem, right? These single-use plastic water bottles wouldn't have to be there if there was a safe, reliable access to clean water for everyone. So I've talked about some unmeasured factors in arbovirology. I talked about solid waste. I talked about the built environment, governance and control programs, and the structural factors that underpin all of it. I want to leave you just mentioning partnership because I can't, like I said, all of this work has been through so many different partnerships. As Steve mentioned, we did spend 5 years with 90 authors around 26 countries of the world writing a book on partnership, which there's a QR code there if you would like to download the book. It's free for all. There are some beautiful key tenets from this partnership book that we continually try to emulate our work, centering trust, expanding the boundaries of science, really disrupting the status quo, working within these imperfect systems, repairing our interconnectedness, and then, of course, experiencing the joy of human connection. And my lab is a great joy for me. I think this is like in Jamie's beautiful newsletter just this week, it is the engine that fuels this kind of work. For students out there, we're going to be teaching a class on equitable global partnerships. If you're interested, on Wednesdays in spring quarter, come find me if you want to talk about that. I always want to thank our amazing study participants who are just the best. And then the students. I don't know if you noticed, I tried to it took forever finding everyone's picture and putting them on the slides because the students have been incredible. Students are really sustainability. I didn't have enough time to talk to you all about all the students. There are many more that were part of this work that weren't represented. And in particular, a ton of our Kenya students, because I didn't talk about all of the vector work today because I didn't have time. And most of our Kenyan students did a lot of the vector work under the mentorship of Bryson and Francis. So to conclude, arboviruses are important, under-recognized, and increasing human health threats in Kenya. Community-based prevention is essential. Research uncovers opportunities for activism and advocacy. The climate and pollution crises have both direct and indirect impacts on health mediated by health inequities. Effective global health practice and research relies on a foundation of stakeholder partnership. And together, we can promote and improve human health and the environment while tackling global health inequities. So I want to thank you for your attention, and I will take any questions.
Questions for Desiree?
Yes.
Climate change is something that we hear a lot about, and when somebody is studying endemic countries to chikungunya and dengue, to the arboviruses, we don't usually think about the United States, but then there was local transmission of chikungunya
in Long Island.
Yeah, exactly.
And there was dengue a few years back.
Yeah, so I just wondered if you have insight onto, is this just an anomaly or something that we anticipate happening more frequently?
Yeah, well, we just submitted a gigantic grant to study local vector-borne disease transmission because we believe that it's just a matter of time. I think that, the world is warming, and there are these inequities that I talk about in Kenya. They're not only in Kenya, they're right here. They're right across the street. And so there are people who are more at risk and more vulnerable and sensitive to these infections. And there's a lot of built environment inequity, right? That puts people at risk, structural inequity. And so these infections have been transmitted along the borderlands in Texas like every year, right? And Florida, which is our Caribbean, right? It's warmer there. There's a lot of movement going back and forth. Again, the way these infections get around the world is not because the mosquitoes fly to Texas, right? It's because humans carry the infection, either knowingly or unknowingly, because again, you can have really a lot of virus in your body where you're infectious to a mosquito that might bite you when you come back home and even have no symptoms, right? And so people move these infections around the world. We saw it with Zika. We saw it with chikungunya when it hit the Western Hemisphere. Chikungunya, Zika, dengue are now happening a lot more in France, Spain, Italy, and so I think it's really just a matter of time, and we will have a lot more of these infections. The one thing that's a little bit different is the way we live. I don't know if you remember my 3 overlapping circles, but human behavior has a lot to do also with risk and risk mitigation, right? So in the US, often in hot places, particularly, people are inside. If they have the means, they've got an air conditioner on, right? Maybe they have screens on their windows. Those do protect, right? But not 100%. And so again, there are people I don't know if you know this, but a couple of years ago, for the first time in, I think it was 20 years, we had malaria transmission in the US. Local malaria transmission, 20 cases, most everyone was unhoused. Yeah. So again, these infections give us an incredible, if you're paying attention, just an incredible perspective to what's wrong in our society, what we need to fix, and where we need to put our attention. Because when we can fix the underlying inequity issues, we fix the disease, Yeah. So, yeah.
So connected.
Yeah, it's very connected. Thank you. Yeah. Any other questions? Come on, ask me about the Ask me about the antibodies. It's crazy, right?
It is.
It is.
I was going to ask you. I mean, I was really struck by all the co-infections.
Yeah.
And you were saying that the initial symptoms are very similar, but
Yeah.
I'm wondering about the disease course, and also its impact on many other things. I mean, the thing that comes to mind is vaccination for not necessarily for those diseases, but just globally. Like how are those co-infections really impacting the health status and immune status?
Yes, totally.
Of people.
So we have a beautiful grant that we've been trying to get funded through the NIH, which keeps getting pulled a lot. And now I think we'll just never be funded, but looking at co-infections, because there is so much malaria and arboviral co-infections. And we've published on this, but our preliminary data shows that, kids that have both, in the end, they recover slower than kids with either. I mean, with malaria, it's amazing, right? Like if I want malaria, I want it in Kenya, right? Because it gets diagnosed really well instantly. The drugs are there and I feel better in a few days, right? So the malaria kids, when they're effectively treated, get better fast. The kids who have dengue sucks. So people feel sick for a while and in a month they're still not fully better, but they're okay, right? But when you have both dengue and malaria, you are sicker, right? And you take longer to bounce back. I think that David in my lab is looking at a lot of the B-cell interactions and more cellular mechanisms underneath what might be happening when you have two infections. And we're really interested in that not seroconverting thing. Because our theory is that malaria might perturb the immune system to respond in a certain way and kind of in a way like almost suppress the immune system so that you are not getting the antibody production that you would normally see. And so that work is ongoing and probably will be published in the next year or so. But interesting and shocking, which means that we have a lot more than we even think we have. Yeah.
Thank you so much for a lovely presentation. So my question, just picking up from where you left off, I was wondering about the role of sickle cell phenotypes, because in malaria, previously malaria, I think there's work from Kilifi showing that having the sickle cell phenotype plus malaria has an effect on immune responses to gram-negative bacteria, especially the non-typhoidal salmonella. And so when those reduction in malaria transmission, those less non-typhoidal salmonella being observed in the populations which had sickle cell phenotype, so that it's a sickle cell disease or the trait. So I'm wondering whether they might be given the distribution of these viruses, whether sickle cell might be a factor. The second question is, I was just looking at the plastics and thinking about wondering whether maybe it's a framing issue about commercial determinants of environmental problems, because I think part of the reason why we have lots of plastics is actually a commercial and a marketing issue, that previously things which would not be packaged. I grew up with soda, for instance, in glass bottles in the same communities, and it's completely switched to plastic. So I don't think it's it might not necessarily just be poverty, but I think also there's definitely a commercial angle to it.
No. Yeah, I didn't mean to suggest that it's all only about poverty with the plastic pollution crisis. It's absolutely capitalism and someone making a ton of money and the pressures on business and greed. So yes. So we haven't looked at sickle cell and interactions on disease with sickle cell. Again, we are a lab that studies malaria by default. I've given talks before to malaria folks, and they're like, "This is a really innovative way to get a malaria study funded." And I'm like, "Yeah, we're doing it because it's so common and so important." But we haven't looked at interactions of at least sickle cell and the way that that disease manifestations might change with these infections. But I did just want to show you, because I couldn't fit it all in, and but Aslam also did look at typhoidal salmonella in kids in Kenya, and this is about to be published. But it was really shocking to find that there was a ton of typhoid on the South Coast, particularly on the South Coast. So there are a lot of other infections that we're not always monitoring for that probably have impacts that we are not detecting in our disease systems, especially when we continue to study them in silos, one infection at a time. Yeah. Miriam. Yeah.
Well, two questions. One, thanks for the excellent talk, and really just looking at the breadth of work. One of the questions that I had was in terms of detection in the community, because the typhoid sort of triggered the question, because everybody who has a fever goes and gets tested, and you all have malaria and typhoid, and you're treated for both. So what are are there any strategies around sort of looking at quality control for testing in regular labs, and maybe has that translated to some of the work you're doing on site? And then the second question was really a more practical question. There's a lot of incredible work, and I think what was truly inspiring was the embedding into the community and the ownership. The question was really around what are some of the strategies that one can employ in terms of really getting that community buy-in, engagement, and ensuring longevity for some of these projects. Thank you.
Yeah, so I think I'll take the second question first. I think, I showed you that picture of Charlie, right? So Charlie had been working in Kenya for 25 years before I got there. And then Charlie's mentor was working in Kenya for 25 years before him. So, and luckily Charlie's a good guy, and Charlie's mentor was a good guy. Okay. So that's the first thing. So I came in on like the shoulders of giants, yeah? Meaning that like people didn't know me, and clearly like if you met Charlie, we are very different people, very, very different. But they like sort of gave me the benefit of the doubt initially. And then I proved that I'm a trustworthy person, and that actually, and I think the way that has happened over time is that like when these studies from the very beginning, right, we like, everybody talks about it now, but co-design, right? Like when Charlie figured out to study Rift Valley fever, what he did was he went to his partners in Kenya and was like, okay, there's this biodefense supplement at NIAID. Like which one of these, it seems important to you? Which one do you want to work on? Which one do you have interest in, right? And this is how everything has happened. And so I think from that way, and then I would say that the people and the scientists and the field workers, everybody that we've been working with in Kenya, they are very connected to their communities. So we've gotten really lucky with working with people who are completely trusted in their communities. So I think the answer is like trust in all the different layers. Trust with us, trust within the community. And I feel like that has really helped us sort of continue and sustain, bring students, bring new partnerships together, and that's sort of the model that we followed. And even now, when this grant has ended, because we can't get any more money from our government because they refuse to fund this sort of work, the partnerships remain. And luckily, we have the nonprofit to sustain those partnerships, right? And we're still doing good work through the nonprofit. Okay, wait, remind me what your first question was, though? Oh, the diagnostics. So we've done some work looking at diagnostics and how horrible they are, at least for arboviruses. I mean, and what is available for like $10 in the private centers, and how it's better to just flip a coin than actually use that. In our work with the policymakers, we work with the policymakers when I go to Kenya, and we talk at the national level, the county levels, and try to encourage, actually, it would be wonderful to actually get testing for dengue in the health centers. There's a lot of competing interests, of course, but I specifically can't talk a lot about diagnostic stewardship or accuracy, because most of all the work that we've done has shown there isn't very much. And so the work that needs to happen is not because the diagnostics don't exist in the world. There are some okay diagnostics, but they're just not very available in Kenya. And the what comes into Kenya for these diagnostics is not optimal, right? And so I think it's high up, sort of, with the private clinics. Like, I think the agreements with these companies, I mean, there's a lot of trash on the market, as you know. And so, yeah, I think it's going to take I think it's going to have to be the government that might step in there and look at accuracy, if they were to actually take on something like this and put it into the government clinics. Yeah.
We are at time. And I want to first give a round of applause for Desiree
Thank you.
for great science and really for inspiring us all to work on these important problems in ways that really build and support communities. So it's just such a gift to have you here at Stanford. And I want to thank everybody for taking the time to come and participate in this conversation. And don't forget, there are lots of great events that CIGH is supporting, and we will keep you informed.
Yes. Check out our events page at globalhealth.stanford.edu or sign up for the newsletter.
Yeah. And if you want the